Director, Dr. J. Rama, respected Head Crop Protection, Dr. Santosh J. and Dr. Sri Kumar, Principal Scientist at ICAR-CRI Trivandrum. Santosh, on this side. Respected heads of various divisions of IASR, dear participants and beloved colleagues. Such an online workshop on art is the brainchild of Dr. J. Rama, our director. Apart from core research, she has always been interested in such outreach programs, not only for farmers and entrepreneurs, but also for students and faculties. Such training programs are regularly being conducted at the Institute with her constant encouragement. I would like to extend a warm welcome to Dr. J. Rama. Dr. Santosh J. actually is the backbone of bioinformatics research at IASR. He is a nematologist and molecular biologist and has developed bioinformatics facilities that we see at the in the campus. He and his team were using this facility and also training hundreds of students and faculties on various aspects of bioinformatics. We welcome you, sir, on this gathering. I would also like to welcome Dr. Sri Kumar, who is the external faculty for this workshop, and all the heads of different divisions. On behalf of the Bioinformatics Center, ICR, IASR, I welcome all the enthusiastic participants. Most of them are students and postgraduate research workers. I welcome one and all to this session. May I now request Dr. J. Rama to release the workshop manual and also invite her to speak a few words.
I declare this manual released. So this has been prepared by our group. So it will be handed over to you on the last day as a soft copy to all the participants. Dear Dr. Sri Kumar, Principal Scientist from CTCRI, our sister institute. We have not met before, I think. Welcome you, sir, to this meeting. Dr. Santosh like what Divya said, he is the backbone of Bioinformatics Center in our institute. So I think you students are really lucky to have two experts who will be teaching you in the next 3 days on bioinformatics, especially on R. So all of you should be very attentive. Now, dear Sona, our bioinformatics scientist. So now ICAR has a discipline on bioinformatics, and last time there were about 11 posts and so in various ICR institutes. So why I said this detailed about her designation is because the students, there are many students attending this, so you should know the opportunities. When you are participating in this workshop, you should also understand what are its applications and what are your opportunities too. So that is why I just said there are opportunities even in agriculture, even in ICR institutes, which is very good. Now, Dr. Divya, she is our biotechnologist who is also well versed. So Sona and Divya are your course coordinators. And all students, I think about 43 of you are there from various institutes doing various courses. So this is a very good opportunity for you. And welcome you to Indian Institute of Spices Research. Actually, this is an institute which does research on spices. All types of research we do, basic, applied, and advanced research too. So bioinformatics is one such area where we are working. And as you know, the applications are numerous. The applications are numerous in various fields like plant science or animal science. Everywhere bioinformatics can be applied. Now, when you look at bioinformatics, if the applications, I don't have to detail you all, but still I thought being a crop institute, a few aspects like in crop improvement, you can use it, biotechnology, microbiology, soil science, anywhere. And in medicines, of course, all of you know, in gene therapy and drug discovery, everywhere it is being used. And as I said, it is a recently emerging field. And for this, you have to have a basic knowledge of many subjects, maybe biology, computer science, and statistics, and of course, mathematics. Everything you should be having a little bit of knowledge, and all those things will be briefed to you when the classes goes on. So I'm not going to detail on all those. And one thing I just wanted to say is that now of late, molecular biology is gaining a lot of importance. And so lot of interest in students to do these sort of work. And because of that, not really because of that, so whatever work we do ultimately, all this should get translated into knowledge. So with the development of technology, as I said, a lot of research is being done. And once it is not that you do the research alone. This research should come out as good publications. So when you do research, a lot of data is being generated, and all this data has to be interpreted in the right way. You have to analyze it, you have to visualize it, and you have to interpret them in the right way so that you get a logical conclusion, good logical conclusion of what you are doing. Then only the knowledge, whatever you do, gets translated to the others with the correct interpretation. So that is where your today's topic, that is on R, helps you. So why R is being used? It is an easy platform where it is easy to work with. So that is the main reason, and it is also available. The rest of the things maybe as the course goes on, they'll be telling you. So with this little bit of introduction, I would request all of you to be alert when the classes goes on, because there is a pre-evaluation test and also a post-evaluation test. That is why the notes are not going to be given to you right now. They'll be talking to you, they'll be evaluating you on your knowledge. And also there are a lot of doubts, you will get a lot of doubts as the class goes on. Please make use of the experts here, because they are all well versed in the field, and get it clarified as and when it comes. Also, you try to understand, because this is bioinformatics. Even I used to get confused whether statistics, biostatistics and bioinformatics, all these are same or not. These sort of doubts would come into your mind. And also, as the classes goes on, you try to understand your opportunities, and also you try to learn the subject well. That's all. And I welcome each one of you to this institute and into this learning process. And also, whenever you have another doubts, you can always contact Dr. Sonash and Divya. They'll be there to help you at any time. Thank you.
Thank you, ma'am. May I now request Dr. Santosh, who is also giving the inaugural talk today, to share his views and wishes on this occasion.
Dr. Rema and Dr. Srikumar, my fellow scientists, and dear participants. It's indeed a great moment for Bioinformatics Center at ICAR to organize one more training program. As you know, I think some of you may be aware that the Bioinformatics Center at this institute has a humble beginning in way back, almost 2 decades back in 2000. And since then, we were trying to develop this faculty, this expertise among the fraternity, ICR fraternity, through a number of training programs. And many of you may be aware, still many of the resources are available online about all these training programs in advanced cutting edge areas like NGS data analysis and other things. So this is a series in this another program in the same this one series, and mostly oriented to the beginners. And we are trying to introduce a new software, the R software, which is quite popular among the worldwide community. So this is the main intention of this training program is to introduce you the open source tool, the versatility of the R tool for biological data analysis. And we have experts like Dr. Srikumar, Dr. Sona, and Divya, who can handle the topic and who can guide you. And we have, I think they have planned a very good layout for the program with a lot of hands-on sessions and other things. Try to make use of the sessions so that you can start. So anything now, only this is only an introduction for this one. So you have to practice. Practice is the only means of no, getting rid of the inhibitions so that you can excel in the area. You can learn new things and you can gain expertise in using these such tools in your day-to-day analysis. I think we will you'll be hearing more about all the applications and the importance of this tool in the coming days. I only wish I wish to compliment Dr. Sona and Divya in conceiving this program, and so we have the momentum is continued, especially with regard to bioinformatics at IASR. I'm happy that we got good successes like these two young ladies who can handle the subject bioinformatics in our institute, which will be helpful to all the related community across the nation. So I once again I'm happy to be part of this program, and I declare that this session is open for declare that it is inaugurated. Thank you so much for the program as well as the invitation. Thank you.
Welcome once again to the training at ISR, the Bioinformatics Center at ISR. So I'll not be going deep into any of these bioinformatics or R or my succeeding speakers will be dwelling more time on it. I know that all of you are interested in bioinformatics and you are because of your interest only you have turned up for such a training program. So we know the bioinformatics, how important it is, and how the changes are happening in biology of late. So today biology, I should say it is digital. So that is the major change happened, maybe during the last 1 or 2 decades. The subject boundaries are disappearing and it is becoming a multidisciplinary subject, not zoology or life science or botany alone. So it's a multidisciplinary subject, which is more inclusive of many subjects. So that is the speciality of today's biology. So that is what makes it more interesting and more maybe exciting. And biology, which was comparatively a docile subject, now suddenly it has become so exciting and thrilling for many of the youngsters. So, and wherever still we are following the traditional courses in biology, there are no takers. And when it is an interdisciplinary or transdisciplinary nature is brought into biology, it suddenly becomes exciting. That's the relevance of biology, and it is started touching many areas in modern life. So that is the importance of biology. And when you say that we know that the whole genome sequences are currently increasing day by day. A lot of data is becoming available. It is piling up, and a number of organisms are now, because it is sequenced end to end. So that is the advantage of bioinformatics. And suddenly the technology has become very popular, maybe cheaper, and every lab, anybody who is working with the biological aspects now is going for the whole genome sequencing. So that way, I think a lot of data is being generated. Have a look at the latest statistics provided by GenBank. On the left side, you can see the number of base pairs already sequenced and deposited in GenBank, and the whole genome sequences. From that, how many base pairs or the nucleotides have been available currently in the database. So you can see there is a very big leap in the sequence information available, which is available in the public domain. You can see when the GenBank contains almost 940 billion base pairs. On the other side, the base pairs or nucleotides data available from whole genome sequences are almost multiple, 13.8 trillion. So that is the difference. Suddenly, though, the whole genome sequence has overtaken the conventional sequencing and other things. And if you compare the number of sequences, also you can see the visible change in or the sudden increase in whole genome sequences available in the public domain. So that shows that we have no dearth of data. A lot of data is getting generated day by day. And what is lacking is information and knowledge. So we are drowning in information and starved of meaningful
Excuse me, sir.
information. Yes.
Sir, we are unable to see the slides. Please
What slides?
put the slideshow mode.
It's not available.
Not visible. Like slideshow mode.
I'm sorry.
It is coming as slide 4 of 33, but we cannot see the graphs and pictures and contents.
Sorry. I don't know. Let me check.
Fine, sir. Thank you.
Yeah. So what I was telling, I think you have missed what I the number of sequences, the available in the genome sequences available in the public domain. No, it is shooting up like anything because of the new technologies, new NGS technology available across the lot of techniques are available, and it has become very cheaper nowadays to generate the sequence information. Many labs are venturing into whole genome sequencing, and this data is currently available. And you, on the other hand, you can see the data available in GenBank, and through the conventional sequencing as well as the whole genome sequencing. You can see on the left side the number of bases available. The whole genome sequences have the data has overtaken the conventional sequencing, and on the other side, the number of organisms number of sequences available also, you can see there's a drastic difference. Hello. And the almost 940 billion base pairs on the base pairs are available through the conventional sequences. And on the other hand, you have a large number, 13.8 trillion base pairs of DNA sequences through the whole genome sequencing. So this shows the sudden momentum in whole genome sequencing happening all around the world. So that is this is the latest statistics as on August 2021. So that is it just shows how much data is getting piled up day by day. So the new biology is just on this. It is all based on this. A lot of data is available, but we are unable to make arrive at meaningful information or knowledge out of this data. So that is what the most challenging task of today for a biologist is to make sense of lots of data available. So any company will provide you the sequences. Unless you sit or some mechanism is available for analyzing them critically and deriving some meaningful information, the whole meaning of the sequencing is irrelevant. So that is what happening nowadays. So unless the concerned scientist is good in analysis, he has the expertise to go deep into the data, what data machines are generating becomes irrelevant and obsolete. So that's what I want to do. So the new biology is this is the sea change what is happening in the new biology. So you have plenty of data available, but you are unable to make derive meaningful conclusions out of this data. So the data is meaningless unless you can generate meaningful information or knowledge out of it. So that is what everywhere now we speak about big data. Big data is available. Big data is analytics is becoming a hot subject nowadays. Not only in biology, any other area, sociology or anything, the statistics or medicine or anywhere, COVID data, everywhere, we are talking about big data availability. So biology is facing same problem. Big data is available, but you have to mine deep into the data to derive meaningful information. So you know that the NGS technologies, how dramatically it has revolutionized the sequencing business. And but when you are finally trying to assemble these short fragments or shotgun sequences, you need the help of a analyst, you need the help of a bioinformatician, you need the help of a computer scientist for properly aligning them, assembling them, and deriving meaningful information out of that. So that is the crux of the problem nowadays. So what is next after collecting data? You can easily collect data, but the post-genomics, handling of this data, that is quite crucial for the success of any experiment or any project. That is what we are talking about today. So the post-genomics operations are mostly after the assembly, how the data should be annotated, how you can compare sequences of different organisms, what are the structural information available through the sequences, what are the functional information available through these whole genome sequences. Such information is called the real post-genomics analysis of the data. So our main goal is to understand the functional networks of a living cell. So unless this data is processed or analyzed in such a manner, you will not be able to arrive at any conclusion. So the data will remain as junk data only. So the role of a bioinformation or a data analyst is quite serious or quite important in the present day biology. And so this is the paradigm shift happening in biology, from the one-dimensional information of DNA through the two-dimensional information of RNA, and you speak about the three-dimensional information of proteins. All of these have very critical role. All of you know that, how important the structure of an RNA, the structure of a protein, and for such information, if you are generating, then only you can arrive at the four-dimensional information of living systems. So a lot of ecosystem, that the same principles are being, the holistic, the ecosystem principles are applied in biology nowadays for anything, whether it is genomics or proteomics or whatever it may be. So this is the importance of data and data analysis in biology. And the simple biological code of nature, the quadruple representation of the genomics or the nucleotide world. So how it is important. So with the help of this sort of understanding, you are able to tamper with the life. You can engineer them, you can edit them, and you can, we are speaking about synthetic biology, writing our own code for life, how you can synthesize life in the lab. So that is the potential of this technology. So when you start understanding the biological data, you are able to manipulate the data, you are able to manipulate life, you are able to synthesize life, and find solutions for the burning problems today our environment is facing, our mankind is facing. So we have seen that how fast we could find a solution for many of the COVID problem or many of the burning diseases where there is no solution at all. A lot of personal medicines are happening. So instead of broad spectrum medicines, now we are speaking about specialized treatments, personalized medicine. So this is all happening just because of our deeper understanding of the life. and customized. You can go to individual based treatment of many of the ailments. So that is the beauty of this new technology. And we know that during the 70s to 2000, the third industrial revolution, where electronics and automation played a lot of key role in developing that revolution, bringing that revolution. So where it was a reductionistic approach from parts, not as inclusive like many other things. Not what you are moving away from the nature. You were alienated. The man was getting alienated from this natural environment, and because of that, this sort of industrial revolutions brought a lot of negative externalities, which has resulted in pollution, waste, and the today's burning problem of climate change. So this is all happened. Now we have learned a lesson from all those things, and we are. So now we are started talking about system science, where this is the other side of the reductionistic science. So where initially we were seeing as from the whole life, from living cell, we will take one part of it, DNA, RNA, or a small fragment of a gene or something, and we are trying to find solutions. No, this has not resulted in much success. So now, and instead it has brought a lot of negative problems and a lot of adverse problems which is now facing the mankind. So because of that, now people started thinking in a holistic terms. So, and this system science, the term was coined way back in 1948 by Norbert Wiener. And now from that, now we have a So now we speak about system biology. So where you have a lot of usage of mathematics to study how genes and proteins interact to produce the complex behaviors of a living cell. So this is the holistic approach. This can be applied to understand anything from movement, energy supply, or signaling, the growth, even the many of the disease problems, cell division, or even how the death, the mortality, all those things we are trying to understand through the system biology. So that is the latest approach, and it is being because of that now we are dreaming of already started the fourth industrial revolution by combining the digital code of computational design and the biological code of nature. Now many things started becoming a reality. It's a symbiosis between organisms, our bodies, our products, and even our environment, even buildings. So now people are thinking of breathing buildings, which can breathe, so as to alleviate the problems of pollution and other things. And you speak about a lot of new technologies like 3D printing, all those things. Now it's all possible by this sort of integration. So the basic thing is we are introducing the ecological principle of circularity into a design. So that is what happening in the latest industrial revolution. So we are learning from the ecological principles to address the problems, the latest ecological problems, whatever is we are facing nowadays. So here comes we were once suddenly mathematics started playing a lot of role in the biology, and we know that because of we were scared of mathematics, we moved away from mathematics and opted for biology. So now such things are going. Initially I told how the boundaries are changing or disappearing, vanishing, and now it is becoming a single subject where a lot of integration is happening. Mathematics, statistics, computational things, and many chemistry, everything is coming together and to understand the life. So that's what happening. So where bioinformatics and computational biology are two fields this has arisen from the growth of bioenterprise around the globe. So these are the offshoots of biology, and a lot of advancements in biotechnology, bioinformatics, and computational biology. It is going to significantly assist the world as it faces the 21st century's impending resource challenges. So we know that resources are becoming very scarce. So we need to address this through by having a fresh look at the biology or life by combining bioinformatics, computational biology. So these all, we may think that these two are different subjects. Bioinformatics and computational biology are two different subjects, very closely related subjects. So bioinformatics is a multidisciplinary field which combines biological knowledge with computer programming and big data. So it is particularly useful when you are dealing with a large set of data, big data, such as the genome sequences I was mentioning earlier. So on the other hand, computational biology, it uses computer science, statistics, mathematics to solve the biological problems. So that is the difference. So one is an application, the other one is a little basic, where you are finding a lot of solutions or a lot of tools you are developing for handling such for problems. So development of algorithms will be there, theoretical models will be there, computational simulations will be there, mathematical models will be there for arriving at meaningful inference. So that is the advantage. That is the difference between both bioinformatics and computation. So both are working in tandem, and where bioinformatics emphasize informatics and statistics. So it is concerned in itself with the gathering and collection of or organization of data. So that's what basically bioinformatics is doing. On the other hand, computational biology, it is trying to develop theoretical models, computer simulations, and mathematical modeling, and for practical application in the biological data, which is available in plenty. So this is what we are trying to say. And so what I want to say is biology. I want to stress upon the same thing, whatever I was telling you earlier. Biology is becoming increasingly computational. So over the last 2 decades, especially after the Human Genome Project, the computational methods have become well established in biology. So we need to sharpen our computational thinking and computational skills, so which are central to the quest of understanding the life in today's situation. So that's what we are trying to address in this workshop also. What are the tools available for such an understanding of life? So computational biology, so what I told, it brings order into our understanding of life. So as Helen Stevens told, biology adapted itself to the computer, not the computer to biology. So that is the difference. So where now we are taking up the statistics, simulation, data management are the core principles of computational biology as well as bioinformatics. So this is very, very important, and it helps and if this is the thing, you may be knowing this person. We should say that this is Carl Linnaeus, the famous botanist, who has started the systematics and taxonomy in botany, and we should consider him as one of the first, the earliest computational biologists, because he tried to bring a lot of orderliness, order in the vague, randomly scattered data in the field of botany. So that's why we call him as the father of systematics and other things. So in that way, all biology is computational biology. Wherever you are trying to bring order by organizing the data, so that you get some meaningful information or knowledge out of it. So that is called bioinformatics. So if that is the definition, he should be called the first computational biologist. So that's what I was telling. So and you can see a lot of organization after the emergence of bioinformatics. You can see a number of databases, quality databases available in the public domain. If you see this is that some of you at least will be familiar with this journal, Nucleic Acid Research, which is a famous journal. And the first issue of every year, they are devoting it to biological databases. What are the databases? New databases available? What are the changes happening or modifications happening in the existing biological databases. A number of them, you can see the growth of biological database. This is the quality and peer-reviewed databases. There are n number of databases available scattered around the different laboratories. Many of the laboratories are bringing out or organizing data. That may not be find a place. This is the peer-reviewed databases available. So the latest figures, the latest issue of the 2021 put the figure around 1647, 1647 databases. You seek anything, any area, any discipline, any specialization, you can find very good number of databases. So suddenly, within 1 year, we have excellent databases on COVID. So around 12 databases internationally recognized, where the community, all the global community is using those such databases for their day-to-day research. So the databases are nothing but the biological data is meaningfully organized so that you can easily retrieve any information out of it anytime you want. So that is the beauty of databases. This is the simple duty of a bioinformatician, organizing the data. This is one of the first and foremost duty of a bioinformatician. And then another thing is the ontology. So the ontologies are just, it's a concept we have imported from computational science or computing science. And to describe the complex problems in anything. And it is actually basically a sociology, the sociology principle, where ontology is, initially it was used in sociology. So now biology, we brought it, and a lot of conceptual framework we are bringing to help us in collecting, organizing, and the publishing the biological data. So that these databases, different databases developed by different sets of people, they can talk to each other. They can share the information. Otherwise, the terminologies will vary, the definitions will vary. They may not be the same data people may be looking at differently. So to bring in uniformity and interoperability, so this sort of ontologies are being developed by bioinformaticians. So this is another duty of a bioinformatician, and how this computational biology is helping us, as a biologist, how it is helping us. So the first and foremost is it lets you see the big picture of a biology. So by introducing statistics and data analysis methods to identify patterns and correlations, we are able to see, for example, how the mutational processes are helping in shaping the genome. What is the effect of a particular mutation? What is its significance? So all these things we are able to visualize and predict based on the bioinformatics, the analysis. And you can align and scaffold the different DNA fragments. I told you the whole genome sequences. First, you are sharing the whole genome into DNA fragments, and later you are reassembling them. So the NGS techniques completely rely on the computational biology for the reassembly of this fragmented genome. So the algorithms, whatever is used now, should be perfect, so that we are aligning the DNA fragments in the proper order, so that you get the correct picture. So this is all very important. So this sort of tools, computational biology tools, are essential to see the big picture of life. Otherwise, the reductionist approach may not help us in understanding life, as I mentioned earlier. So we need to see in totality, the big picture of life. Then only we can arrive at meaningful solutions for any problem. So another thing is the computational biology provides an atlas of life. So we know that the map, the reference map for anything, it is a reference map, how holistic it should be. So by combining this sort of large data sets, we are able to provide a reference map for biology. We know that it's a low resolution, still a lot of information is hidden. We are yet to bring out all the complexities of life, but still something, even if it is some parts are missing, it is an indispensable tool for biologists. So you know that now any sequence, you have a whole set of data available in GenBank to compare with. So that is the beauty of it, and it forms the background for planning our future experiments. So this is whatever information available now, we are planning our future set of experiments. even if it is a small scale experiment or big experiments, this sort of reference map is important for. So computational biology helps us in providing this sort of a reference tool, a reference map for our the holistic map for our biological process. Another thing, the third and final aspect is the biology, the computational biology is turning our ideas into hypotheses. So we are able to visualize many things. We are able to dream about many of the complexities of life by the whatever information already available. So it really reshapes the biology by making fuzzy concepts rigorous and testable hypotheses that we are generating, and finally we are testing them in the lab, in the wet lab, and to see that whether it is correct or whether it is wrong, or what we should modify it. So this so we are able to plan a lot of new experiments in the in silico environment. So finally only we are going to the wet lab and validating them. So that is the advantage of computational biology. So there are n number of tools for anything. No, you have from homology and similarity tools, like a simple blast to anything. No, protein function analysis, structural analysis. So you name anything, you have tools, plenty of tools. Even a small program like problem like primer design or phylogeny, you have thousands of software tools available nowadays in the open domain, public domain. So you have anything, no, DNA sequence or RNA sequence analysis, protein sequence analysis, enzymes, diseases, gene expression, immunological databases, like that, no, genomic databases, proteomic resources. You have plenty of tools now available. Again, now coming to the nucleic acid, the July issue of every year now, nucleic acid research journal brings out a special issue dedicated to web-based software resources for analysis and visualization of molecular biology data. So this is a quick reference for our many of our things. When we are, no, we don't know when some new analysis you want to take. Have a look at the already available tools. Many of them may be useful for us. We need not research and find out a new tool or develop a new tool for that. Many of the things may be already available. Again, another new compilation. It is available called Aviator. It's where the entire sets of bioinformatics tools are listed and as availability, their link, who has developed it, what was the journal or the article. So like that, no, all information is nowadays available and compiled. No, you can go through that. You can search for your requirement and design your experiment. You can analyze the data using some of these tools are already available in the public domain. So this is the beauty of bioinformatics, where no, it is globally available. Anybody can share their expertise. You can there is no restrictions available. So it's the information is free flow. So and the databases like GenBank and other things, no, whoever they have compiled the information available across the globe, and freely it is accessible for anybody, so that our research, so never know it will be impeded. You can design your experiments. You can find out solutions to your problems by using the data or generated by somebody else. So that is the beauty of this. Bioinformatics has brought all the scientists in one platform, and we are sharing our whatever we are acquiring, and whatever data we are generating. So it is all available at one place for the community to exploit. So that is the advantage. So here again, we have a lot of open source tools, proprietary tools. You have free tools as well as paid software. So this, what are the differences? Again, there are companies to make money. Though they are called the proprietary software. They provide you attractive graphical interfaces, and they combine and very good several softwares and bring out as a pipeline. So the only thing, you just feed your data, it will provide you beautiful information at the end of the pipeline, and you get excellent charts, everything. This is all very very attractive. And only thing is, you don't require any programming skills. Anyway, it's quite user friendly, and always you have a technical support from the company people. So this is all the advantages of the proprietary softwares, but they are exorbitantly costly. You know that CLC or Schrodinger, no, every year you have to spend 15, 16, 17 lakhs for a license. So whether it's not affordable to a common person, common scientist or biologist working in any lab. So, and you don't have the flexibility. You cannot meddle with their code. So it's a rigid thing. No, you have to put your data, you get the information or the results. But on the other hand, you have hundreds of open source softwares where the source code is free. And if you have a little knowledge of programming and other things, you can play with that. You can modify that the source code available. You can customize it to your requirement. And same, it's very powerful, and you can generate equally good results by using such open source tools. So, and open source communities are there, so you can chat with them. There are a lot of resources. You can exchange ideas with them. Always there are people to help you. That way also some community support is available. So this is the difference between open source and proprietary softwares the currently available. And there are programming tools available in bioinformatics. You have whatever available generated by the computational group, computing scientists. Now we have adapted to our biology environment. You have a BioPython, BioPerl, BioJava, Bioconductor, where the today's topic R is being used for biology data. So this is all plenty of tools. If you pick up some of these skills, you can do wonders in biology. So that's what I want to. So a new species of biologists is emerging, where you have the skills. You are acquiring the skills in data analysis. You are acquiring the skills of programming, so that you can, as you wish, you can analyze your data. You can go deeply mine into the big data available with you, and come out with excellent, exciting results. And so what I advocate is, you have to enhance, you have to raise up your proficiency bar, and you have to be continuously on a learning mode. You have to learn and unlearn and learn new things, because it's a very dynamic field. A lot of new things are happening, emerging every day by day. So so what we need, the need of the hour, is a computationally literate biologists. So the conventional biologists are, they are still, difficult to understand the new order of biologists, who are very good in computation analysis and other things. So the traditional biologists, they believe only in wet lab. So here now we are coming out the new brand of biologists who are doing the experiments in the in silico environment first, then finally only they are going to the lab to validate them. So this approach, many the old set of people, like us, we fail to understand them. So we don't immediately recognize them. We don't accept them. This is all happening, but the new species of biologists are now started dominating, and a lot of new things are coming out with their help. And so what the three essential skills for the new set of biologists are, modern biologists are: the first is the command, second is the program, and third is the data. So what you need is, you have to understand how the Linux environment, how the Linux commands or Unix commands work. That some fairly good understanding should be there. You should know the programming. You should have the programming skills. Any of these the programming languages, either Python or Perl or R or whatever it may be. So you need to skill up yourselves. And of course, finally, the data management, data analysis. You should know some of the how to develop a database, how to go deep into the database, how to mine information out of database. So these are the three core skills today's biologist should have. And coming to so this R is again, we have seen R is again an open source tool, free tool, which is now used by not only biologists, so many people are using it, statisticians, everywhere. R has now is a very powerful scripting language for, and we have the biologists have embraced it. especially the sequence analysis or anything. No, I think I'm not going into the details. The coming sessions will tell you more about the art. So it's a well-designed and efficient, widely adopted, and where a lot of a big community is there to help you out. So a lot of flavors of R are now available, like many new versions are available. So this is all possibilities, and this workshop is essentially conceived to introduce you the essential ideas and tools of R. And so we will be guided. You will have plenty of most the hands-on sessions, and of course we know that we have only 3 days, but I think that will introduce you the art environment, introduce you to the art environment, which you can continue to practice and really gain some good expertise in the coming days. So that's it. I think I'm concluding here. Thank you for your patient listening. Any question or anything, I will be whatever possible, I will be able to answer you. So wish you a happy learning. Thank you.